Hello, and welcome to my webinar on Is Your PCR Reaction Healthy? My name is Catherine Meckling, and I am a Technology Development Molecular Workflow Scientist for MilliporeSigma. MilliporeSigma is the life science business of Merck KGaA Darmstadt, Germany, and operates as MilliporeSigma in the US and Canada. Today's webinar, I will start by breaking down the building blocks and mindful actions scientists need to make in order to have robust PCR reactions. Then we will look at sample purification workflows and their upstream impacts on PCR. I will be highlighting workflows for genomic DNA isolation. And finally, I'm excited to introduce you to a new technology from MilliporeSigma called GenElute E Single Spin technology. You may be thinking, what does health have to do with my PCR reaction? Well, you may also ask yourself, what makes something healthy? Health is the state of a physical condition which is absent of impairment. And with PCR, when things go wrong, they go wrong real quick. So let's look at what makes up a PCR reaction to ensure a good foundation of preventative actions for reaction optimization. The polymerase chain reaction, or PCR, has become one of the mostly widely used techniques in molecular biology. It is used in applications from basic research to high-throughput screening. While it is a powerful technique, the universal adoption in diverse range of applications is due to its apparent simplicity and relatively low cost. The technique is used to amplify specific target DNA fragments from low quantities of source DNA or RNA, for which then there's additionally needed a reverse transcription step to produce complementary DNA. One major advantage of PCR is that target sequences can be amplified from a single copy of starting material, even when the template is degraded and contaminated with inhibitors. However, that's not always the best case. PCR consists of cycles of reactions of heating and cooling. Each temperature plateaus is used to control a defined stage of the reaction, and the incubation times are dependent on the instrument reaction plates or tubes or reagents themselves. These should be optimized for each experiment, especially if increased detection sensitivity is required. Each cycle consists of 3 key stages, such as denaturation, annealing, and extension, and each reaction consists of key components such as the nucleic acid template, primers in the forward and reverse direction, a polymerase enzyme with its preferred reaction buffer, and nucleotides, plus some optional additives to help boost up that PCR. When preparing a PCR, a scientist will ask themselves several questions, such as how much template do I need? Are my primers specific enough for my target sequence? Do I need a high fidelity enzyme for this reaction? How many nucleotides and what concentration do I need to add to the tube? And also, are there other contributing factors that I don't see that can lead to the success of my reaction itself? Most importantly is to look at the template itself and not the DNA we expect to see, but the reality of the template, which can be a complicated inquiry involving primary, secondary, tertiary, and even quaternary structures. Therefore, the integrity of the initial DNA template is crucial for PCR success. Some parameters that should be considered when PCR performance is low is ensuring that a polymerase is not sterically hindered. The enzyme is only reactive at specific temperatures, which will help to prevent primers from mispriming, as well as having sufficient target sequence available to the enzyme itself. Low amounts of template, for example, less than 10 nanograms of human genomic DNA, will require specific reaction modifications, such as changing the cycle number or redesign of the primers, or also using hot start enzyme. Also, it is essential to have accurate quantitation, otherwise you may be blind to factors impacting the reaction. There are several factors that impact templates quality. For instance, large amounts of RNA in a DNA template can chelate magnesium ions and reduce the yield of the PCR. Also, impure templates may contain polymerase inhibitors that decrease the efficiency of the downstream reaction. Beyond contaminants, template DNA should be of high molecular weight, and it's recommended to check the size and quality of the DNA by running an aliquot on an agarose gel or checking it on a fragment analyzer. Always, when testing a new template, a positive control should be included with primers that amplify a product of a known size and produce good yield. The amount of template in a reaction strongly influences the performance of the PCR. The recommended amount of template for standard PCR is a maximum of 500 nanograms of human genomic DNA, or 1 to 10 nanograms for bacterial DNA, or 0.1 to 1 nanograms of plasmid DNA, and each sample type should be optimized by the user. In most PCR applications, it is the sequence and the concentration of the primers that determine the overall assay success. For convenience, several primer design software programs are available. Primer sequences have the following general characteristics, such as 18 to 24 bases long. They don't have any of those pesky internal structures that can cause steric hindrance. They're made up of about 40 to 60% GC rich areas, and there's a balance between GC and AT rich domains. There's no complementary binding to each other at the 3' ends, which can cause primer dimers. Additionally, it's very important in regards to the melting temperature itself for annealing temperatures. Typically, they're between 55 and 65 degrees. However, you can use higher melting temperatures for better specificity, such as between 62 and 65 degrees. Of note, optimal annealing temperatures are often higher at higher melting temperatures of the primers, between 5 and 10 degrees higher than melting temperature, and they need to be determined empirically. For both primers, the melting temperatures are typically similar to allow for the reaction to be homologous. The primer concentration between 0.1 and 0.6 micromolar are generally optimal. A higher primer concentration may promote mispriming and accumulation of nonspecific product. Lower primer concentrations may lead to an exhaustion of the enzyme before the reaction is completed. This could result in lower yields of the desired product. Additionally, of note, for some systems, a higher primer concentration, such as up to 1 micromolar, may improve results. And when testing new primers, again, always use a positive control reaction with the template that has been tested for a functional PCR, and this control will show that whether or not the primers are working. Shown here is a gel electrophoresis image of an assessment of different reverse primers, whether they are by themselves or with the forward primer in the reaction. In the image, you can see that even without a forward primer, some PCR product could be produced, and that there are additional bands which can indicate that their primers are mispriming. Sigma has several tools associated with primers themselves, such as the Oligo Evaluator, and you can order oligos directly from Sigma themselves. Polymerases are the PCR athletes of every reaction. Just like how a long endurance runner is different from a speed racer, the choice of a DNA polymerase can profoundly affect the outcome of a PCR. For most routine PCR, a Taq DNA polymerase has long been the standard PCR enzyme. However, Taq DNA polymerase has its limitations. For most assays, the optimal amount of thermostable DNA polymerase, or a blend of polymerases, will be between 0.5 and 2.5 units per 50 microliter reaction. Increased enzyme concentrations sometimes lead to a decrease in specificity, and this is typically to say like there's too many players on the field. There are several features that differentiate polymerases. For example, one polymerase may be a hot start, meaning that the extension does not occur until after the primers anneal due to the elevated polymerase activation temperature, and using one of these will definitely help prevent a lot of the mispriming. They could also have proofreading capabilities in both the 5' to 3' direction and or the 3' to 5' direction. And this indicates that they're high fidelity. They can be optimized to work for multiplexed PCR reactions with multiple targets on the same template, meaning they can work with multiple primers. Long PCRs require endurance for long amplicons, and fast PCRs work more rapidly at incorporating the nucleotides themselves. Additionally, some templates have high GC content and require the ability to work in a high temperature reaction without the polymerase's denaturing. Also, for convenience, the polymerase may or may not come with a master mix with everything needed for amplification without additional additives needed. Sigma does have a PCR selection guide on our website to allow for you to choose based off of whatever type of polymerase you may be needed. So I just wanted to indicate this here. Sometimes a master mix is not always available and other additives are needed, such as magnesium ions form soluble complexes with nucleotides, and they produce the actual substrate that the polymerase recognizes. The concentration of free magnesium ions depends on the concentrations of compounds that bind the ion, including nucleotides, free pyrophosphate ions, and EDTA. For best results, always determine the optimal magnesium ion concentration empirically, and the optimal magnesium ion concentration may vary from approximately, for a good range, is 1 to 5 millimolar. The most commonly used magnesium ion concentration is 1.5 millimolar. And this is when the nucleotides are at a concentration of 200 micromolars each. Magnesium ions influence the enzyme activity and also can increase the melting temperature of a double-stranded nucleic acid. Excess magnesium ions in the reaction can increase nonspecific primer binding and increase the nonspecific background of the reaction. Always use a balanced solution of all 4 nucleotides to minimize any kind of polymerase error rate. An imbalance of nucleotide mixtures will reduce attack DNA polymerase fidelity. Of note, you can increase the concentration of nucleotides. When you do that, though, you must also increase the magnesium ion concentration. Increases in nucleotide concentration will reduce the free magnesium ions, thus interfering with the polymerase activity and decreasing primer annealing. The final nucleotide concentration should usually be between 50 and 500 micromolar for each nucleotide. And the most, as already indicated, the most common nucleotide concentration used is 200 micromolar. Additionally, you want to think about the pH, and generally the pH of the reaction buffer supplied with the corresponding thermostable DNA polymerase is between a pH of 8.3 and 9, and these will give you optimal results. However, for some systems, raising the pH may stabilize the template and enhance results. In some cases, adding the following compounds below, and there's a big list of them, can enhance the efficiency and specificity of a PCR. But however, I won't go into depth into many of those. I just wanted to let you all know that they exist. There are some tools specific from Sigma that are PCR additives that are used at different steps to prevent mispriming during PCR. The thermostop additive improves PCR reaction specificity and yield, resulting in a cleaner amplification. Thermostop, Thermogo, and Thermostop RT reagents are novel, easy-to-use PCR additives that act at different temperatures, unlike hot start PCR. Thermostop and ThermoGo additives suppress errors before, during, and after amplification for reactions starting with either DNA or RNA, therefore enhancing PCR specificity and sensitivity. These PCR additives may be multiplexed in diagnostic reactions for more accurate and informative assays, and they can also enhance the quality of downstream applications such as next generation sequencing, deep sequencing, and Gibson assembly. The Thermostop RT additive is also a double-stranded nucleic acid. However, it acts as a hot start for reverse transcriptase during the one-step RT-PCR as well as two-step RT-PCR. And that's coming from RNA samples. Now that we are all on the same page of what goes into the PCR action, let's look at how everything is put together. In an optimal reaction, less than 10 template molecules can be amplified in less than 40 cycles to a product that is easily detectable on a gel stained with lithium bromide. Most PCRs should therefore include only 25 to 35 cycles. As cycle number increases, nonspecific products can accumulate. Shown in this graphic is a purified or unpurified isolated nucleic acid template at cycles numbers 40, 60, and 72. As shown, there are nonspecific products and aggregated products starting to accumulate, and this makes the PCR product less homologous. Also, keep in mind that errors can occur due to thermocycler-based problems such as inconsistent ramp rates and well-to-well temperatures. Always ensure that your equipment is working correctly. It is essential to denature the DNA template completely while going into the PCR. Initial heating of the PCR mixture for 2 minutes at 94 to 95 degrees is enough to completely denature a complex genomic DNA so that the primers can anneal to the template as the reaction is cold. If the template is only partially denatured, it will tend to snap back very quickly, preventing efficient primer annealing and extension, or leading to self-priming, which can also lead to false positive results. Denaturing during cycling at 94 to 95 degrees for 20 to 30 seconds is usually sufficient, but this must be adapted for the thermocycler and the tubes being used. For example, longer times are required for denaturation in 500 microliter tubes than in 200 microliter tubes. If the denaturation temperatures is too low, there will be incomplete melting of the DNA and they'll snap back as described earlier, thus not giving access to the primers. Use a longer denaturation time or higher denaturation temperatures for GC-rich template DNA, as they will have a higher melting temperature. Of note, never use a longer denaturation time than absolutely required for complete denaturation of a template DNA. Unnecessarily long denaturation times will decrease the activity of attacked DNA polymerase. For most purposes, annealing temperatures need to be optimized empirically. The choice of the primer annealing temperature is probably the most critical factor in designing a highly specific PCR. If the temperature is too high, no annealing actually occurs, but if it's too low, nonspecific annealing will increase dramatically. Primer dimers will form if the primers have one or more complementary bases, so that the base pairing between the 3' ends of the 2 primers can occur. Attacked DNA polymerase can add approximately 60 bases per second at 72 degrees Celsius. A 45-second extension is sufficient for fragments up to 1 kilobases. For extension of fragments up to 3 kilobases, allow for about 45 seconds per kilobase. However, these times may need to be adjusted for specific templates, such as ones that vary with GC content. For improved yield, use a cycle extension feature of the thermocycler. For instance, perform the first 10 cycles at a constant extension time, like the 45 seconds for 1 kb product, and then for the next 10 cycles, increase the extension time by 2 to 5 seconds per cycle. For example, 50 seconds for cycle 11 and 55 seconds for cycle 12. Cycle extension allows for the enzyme more time to do its job because as PCR progresses, there is more template to amplify and less enzyme due to denaturation during the prolonged high PCR temperatures to do the extension. As usual, after the last cycle, the reaction tubes are held at, for example, 72 degrees when using a DNA tag polymerase for 5 to 15 minutes to promote the completion of partial extension products and annealing of single-stranded complementary products. There are some differences if we take PCR to the next level. For qPCR, for example, you can do a cyber-based assay or a probe-based assay. For cyber, it's highly specific to a double-stranded DNA, and this is performed using a dye. It monitors the amplification of the double-stranded nucleic acid sequence. Additionally, another advantage is that there's no probes required with this assay, and this helps to reduce the initial setup needed and additional running costs associated with it. There are multiple dyes that can bind to a single amplified molecule, and this increases the sensitivity for detecting amplification products. The typical cost for a reaction is typically very low. However, some of the drawbacks of using a cyber-based qPCR assay is that it's not specific, for example, to a target sequence, and it can also lead to false positives because of it's not a very specific assay. However, you can use a probe-based detection using a fluorogenic probe, and this is very specific to a target sequence. These assays also typically have a very low background, and this is non-specific fluorescence, so there's a greater chance of having fewer false positives. The label probes are very different, and they have distinguishable reporter dyes and allowing for amplification of 2 distinct sequences in one tube, so you can multiplex as well. However, because of the specificity of this, there is a different probe required to be synthesized for each of the target sequences, and this can really add up in cost over time. So you have to do a balance when deciding what type of qPCR assay you're trying to perform. Looking a little bit deeper into qPCR itself, there are several key components of a qPCR when looking at the data itself. There is a baseline, and this is where there's no reaction will occur, and this is typically added to by using a mock sample where there's no reaction taking place. So typically, and the enzyme itself won't be added, and so the dye will intercalate with the double-stranded DNA. However, it will not increase because it isn't forming additional and amplifying. The fluorescence is indicated by the Y axis, and this will be essentially brought about by a dye or reporter that probe-based assay. The cycle number, which is along the x-axis, will tell you essentially grow as each cycle takes place. This is the number of amplification reactions that are occurring during the PCR. The plateau is the level at which the instrument can no longer differentiate. the fluorescence. So essentially, this is very similar to an overexposed sample on a gel. The exponential phase is the increasing fluorescence of a sample from the baseline. And this is essentially showing you as the number of products being added, the fluorescence will increase. There is a threshold indicated as well, which will essentially allow for you to differentiate your sample from the baseline itself. And every qPCR will essentially cross that threshold at a specific CT or CQ value, which will tell you that the sample is being recognized as being amplified. There are other forms of PCR that scientists use, such as RT-qPCR for RNA samples or digital PCR to determine copy numbers of DNA per volume. However, keeping the discussion more broadly to PCR, let's look at some of the common pain points and possible causes associated with PCR itself. Some of the common pain points associated with PCR is that obviously there's no band being produced when you're looking at it under gel electrophoresis, or there's no, your sample isn't crossing that threshold on a qPCR, indicating that the amplification isn't happening. This has so many different factors associated with it, such as, again, those magnesium ions, which affect the polymerase, the amount of template you're adding, as well as mispriming of the primers. So there's several key contributing factors for each of the pain points associated with PCR. Another key pain point is misincorporation of nucleotides, or that the enzyme itself is low fidelity, so it doesn't really match your initial template. Additionally, there could be those nonspecific bands typically associated with the primers themselves, or there can be smeared bands, which usually means that there's product being produced of different sizes, so the PCR isn't being able to be completed. Additionally, low yield can also indicate that the primers are either mispriming, or that the cycle number could be too high, that the enzyme activity is too low. There's several factors that come into play when troubleshooting a PCR, and to say the least is a complex troubleshooting question and requires a lot of backtracking when the reaction fails. That is why it is always beneficial to understand the building blocks of the reaction to provide more insight into solutions when the reaction breaks down. Although the building blocks of PCR are contributing factors to its success, so are the upstream factors from sample preparation. For example, some PCR inhibitors are common from specific sample types, such as humic acids from soil or plant tissues, or hemoglobin from blood samples. Additionally, PCR inhibitors that can be commonly introduced during the experimental processes of the sample isolation itself, and these can include tris, ethanol, isopropanol, EDTA, even the enzymes themselves, which will be protein, and then a guanidinium isothiocyanate, which is a cheotropic salt, as well as a phenol, which is an organic solvent if you're doing a phenol extraction. And as previously stated, the template is very important since it is the starting point from which amplification begins. So let's look at sample preparation and how the upstream can impact the PCR. After sample collection, there are 5 key workflow decisions a scientist makes regarding their sample preparation. The first step is homogenization of the sample. Mechanical disruption by using a pestle or a bead beating is often used for tough sample types, such as animal or plant tissues. However, samples like cells or blood can be pelleted before lysis. The next step is breaking open the sample and digesting any unwanted biomolecules through lysis. For nucleic acid sample preparations, typically a lysis buffer is added that will assist with breaking into the cells. Proteases, like the ubiquitously used protease K, are used to digest the proteins that can degrade nucleic acid. Optionally, if assessing for DNA, sometimes an incubation of RNAs will help to remove any RNA, and that will be performed. Or if RNA is being assessed, a DNase is used to help digest any DNA in the sample. Purification is the choice of method for separating and isolating the biomolecule of interest. And there are several ways of performing this process, and I'll dive more into them as we go on to more of the more common workflows later in this presentation. The next step is modification, and it is the process of making changes to the nucleic acid that allows for the final step, which is assessment. There are now methods where scientists will amplify their nucleic acid by PCR directly after lysis. However, for unbiased assessment results, a purification is still recommended and required. Most scientists group lysis and purification together for sample preparation, and this is because there are a lot of off-the-shelf products that exist to address these needs. But before I go deeper into the different methods of purification, it is important to think about some of the key pain points when thinking about sample preparation itself, and that can impact downstream sample modification and assessment, as previously discussed. The two key drivers for downstream failure is a poor quality and poor yields, and a lot of this is due to inherent variability of the sample. This can be between different sample types or within the sample itself. Oftentimes, a user will need to perform optimization assays to validate for their sample type. A key driver is that there could be some incomplete lysis, that the cells aren't even opening up to allow for the DNA template to be accessible. Another contributing factor to yield is the fragment length itself. And this is, the DNA will degrade over time due to potentially like those DNases within the cell. Additionally, there's some carryover process contaminants, such as binding salts and organic solvents, that can have detrimental impacts on downstream processes. And it's very important to keep those factors in mind. One of the most common methods for nucleic acid isolation is using silica spin columns. This is widely used out of convenience, where commercially available kits can be performed safely without a fume hood. The silica membrane is positively charged and therefore easily binds the negatively charged nucleic acids. Again, chaotropic salts are there to assist with the denaturation of proteins from the nucleic acid, and they also assist in the binding steps before the sample is washed with an ethanol solution. And so essentially this began an age of for sample preparation of a bind wash elute. Another common method is using magnetic beads, which is very similar to the silica columns, where silica magnetic beads are positively charged and therefore easily bind those negatively charged nucleic acids. Yet again, chaotropic salts are there to assist in the denaturation of proteins from the nucleic acid and also assist in the bead binding steps of the sample to the bead before the sample is washed with ethanol solutions. And finally, it is eluted in a stored buffer. This process is widely adopted due to its ease to be automated over silica spin columns. Thus, it makes it very popular with high throughput labs that process several samples at one time. I hope by now you've all noticed a pattern, whether it's phase separation and cleanup, silica magnetic beads, or silica spin columns. The process can be very tedious with binding and washing the nucleic acid. The tube handling can be prone to human error, and these methods typically run the risk of cross-contamination. Yet what if those bind and wash steps could be eliminated from the workflow? Now I get to introduce you to the GenElute E Single Spin technology, which where the purification step happens in a single spin. This is done by eliminating those bind and wash steps, and this definitely helps with reducing the number of tube handling steps, and also helps to promote for more efficient and safer sample handling. So what is negative chromatography and how does it work with single spin? So I'll go into that in the next few slides. You may be thinking you cannot just eliminate the bind and wash steps. However, by using a negative chromatography method, it is possible. Instead of binding the nucleic acid and running the risk of insufficient binding or nonspecific binding of your sample, negative chromatography uses size exclusion to fractionate the components of the sample by size. This eliminates several process biases associated with binding nucleic acids to a membrane. Larger biomolecules like nucleic acid flow more readily through a resin bed, while smaller contaminants are absorbed and retained by deferring the migration speeds through the resin beads. And the contaminants will take a longer time to make it through. As you can see in this diagram, kind of think of it as using your GPS, where if you want to take the scenic route, it takes a lot longer to get there. However, you can use the highway, and that will get you there a lot faster. So think of it as slowing it down by increasing the route in which the different biomolecules have to make it through the column. Clearing the sample and packing the resin before loading a supernatant will prevent a cellular debris from eluting in the final sample. These workflow changes provide the three key advantages over silica, which is the simplified workflow, the superior performance, and the reduced waste because of eliminating a lot of the tube handling steps. And best of all, there's no cheotropic salts or ethanol. And when considering a size selection, it is important to know that the molecular weights of the different components of the sample, what fraction you're trying to collect, and how specific you can be to the target biomolecule versus other potential molecules of the same size. Chromatography is a very similar format to using silica columns. They use similar equipment. And it makes it very easy to transition between the two technologies when comparing them side by side. Insufficient lysis can be a setup for failure from the start, and that's why we took a more targeted approach with GenElute E Smart Lyse Protease mixes. These reagents were chosen for two specific reasons. One, their efficiency to work in a non-cheotropic environment, and two, their optimization for their sample type. For example, animal tissues and bacterial cell culture samples require totally different lysis strengths. Using a sample-specific approach helps to cut down on incubation times, and no cheotropes in the lysis buffer means no cheotropes in the final sample. Additionally, preparing the spin column for purification can be performed simultaneously to the step for even more workflow time savings. So let's take a closer look at the column itself. Preparing the column for use is a simple process. The sample collection tube is used to collect the void volume of the column and can be reused since it never actually touches the sample. Additionally, the cap can be loosened a quarter turn, or a cap punching tool can be used to prevent vacuum from forming within the column. You need to snap off the bottom closure of the column to ensure that the sample has a place to go. Additionally, before spinning, the beads need to be resuspended to ensure that the resin beads pack evenly and you're getting the fraction that you need. And then your column is ready to be used to be loading your sample for final purification. Additionally, with this product, you can now choose your own workflow. You can now load your sample directly into the column without opening the purification column. All you need to do is use the GenElute E Single Spin Cap Puncher device. This helps to reduce cross-contamination by keeping the sample contained between transfer steps. While this tool is meant to be used Specifically with GenElute E, purification columns is not required for purification. You can load your sample directly onto the resin bed. Just remember to keep the cap loosened a quarter turn if no hole is punched into the cap. One spin and you're done, and now you have GenElute E purified nucleic acid. Nucleic acids have traditionally been quantified by measuring UV absorption using a spectrophotometer. The absorbance of a diluted sample of DNA or RNA is read at 260 nanometers and 280 nanometers. The optical density, or OD, at 260 nanometers is used to determine the RNA or DNA concentration in a sample using the Beer-Lambert law, which predicts a linear correlation between absorbance and concentration. An OD at 260 nanometer reading of 1 is equivalent to approximately 40 micrograms per milliliter of pure RNA and 50 micrograms per milliliter of pure double-stranded DNA. However, the Beer-Lambert law is only valid for OD readings up to 2, and the stated OD concentration relationship relies upon the samples being pure. Evidently, contaminating substances with absorptions at 260 nanometers or 280 nanometers will impact the estimated OD reading. Pure RNA has a 260-280 of about 2.1, whereas pure DNA also will have has a different 260-280 reading at 1.8. Additionally, what the sample is diluted in may also impact 260-280 ratios, such as DEPC. Water from a pH of 5 to 6 will have an expected 260-280 at 1.6 nuclease-free water. By increasing the pH from 6 to 7, will also increase the 260-280 ratio to 1.85. Storage buffer like TE at a pH of 8 will have an even higher 260-280 ratio at 2.14. And that's very important to keep in mind when calculating your sample yield. Additional contaminants may also impact the ratios, such as phenol contamination, a high protein contamination, and those K-tropic salts, a guanidine isothiamate concentration will also impact the ratios. So let's look at how different contamination will impact based off of different contaminants. Such as tris being in your sample, there really is no significant influence of the absorbance of the spectrum of RNA and DNA associated using UV spectrophotometry for downstream applications. Ethanol or isopropanol contamination, when measured in a storage buffer, will not have significant influence. However, if it's measured in pure water, it will essentially impact it. It will lower the overall 260-280 ratio than 2. And seeing this will also be an indicator that ethanol or isopropanol is within your sample. Additionally, protein contamination, when the concentrations are high, you can be able to detect this in the 230 range, and this will shift the 260-280 ratio. However, at lower protein contaminations or moderate, you really can't see an impact on the results. Again, indicated before, the K-tropic salts will have a small effect on the 260-280 ratio. However, at higher concentrations, they will also impact it at higher rate. And this will dramatically drop the 260-230 ratio. So when you see a dramatically drop 260-230 ratio, this is typically an indication of process contaminants from the actual sample preparation itself. Additionally, the K-tropic salts can also impact downstream assays, so it's very important to keep those out of your sample. Finally, phenol contamination can also very much impact your downstream applications because this will be measured at the 270 nanometer wavelength, which will definitely shift that 260 that DNA and RNA concentrations are being measured at, especially for any kind of concentration below 10 nanograms per microliter. So it's very important to keep phenol or any large heavy phenol organic solvents out of your final sample. Remember those organic solvents and cheotropic salts. So we're going to look a little bit more into them more in depthly. So by comparing the two technologies of a silica-based assay versus a GenElute E Single Spin, you can definitely see, or you could potentially see some differences in the samples when viewing under the UV spectrophotometry. So especially in that 230 range, because again, those cheotropic salts are showing up. So if you're looking at these, essentially what you're looking for is this, so that the teal and the yellow line should match. This indicates that there are no process contaminants because they match the baseline wavelength or the baseline that you're using. A pure sample in red should also match what's coming off of the column, which is purple. However, you see with silica that you're seeing all these process contaminants that are added together, which causes this tail at the 230 range. And this is important because this can impact those downstream quantitations and also impact downstream assays themselves. And have you ever seen your sample float out of the well of a gel? Well, this will likely be due to remaining ethanol in the sample after purification. This can be remedied by diluting the sample. However, this may also be limiting to your detection of your gel electrophoresis because you're not able to load as much within the well because the well may have a certain volume capacity. There is no ethanol washes in the GenElute E Single Spin technology, so gel electrophoresis can be more successful. Spinning the sample only once also has other advantages, such as higher genomic DNA. And since this is a size selecting technology, you're eliminating a lot of those shared contaminants. Additionally, shown here is essentially 4 replicates from a silica-based and 4 replicates from GenElute E. So you see that there's more homology coming from a GenElute E column versus silica-based. There's differences between each replicate, and there's a lot more being up at a lower molecular weight, these smaller fragments. And those smaller fragments are essentially being added to the overall quantitation from a UV spectrophotometry data, so you're not loading as much. So just think of it as this is what you're adding into your downstream assay. You're not adding the full-length, high molecular weight DNA that can be critical for those downstream applications, such as next-generation sequencing that need long fragments like PacBio, or electron microscopy. Additionally, the silica-based can also, just the whole workflow itself can actually shear the genomic DNA leading to those smaller fragments. So it's always a good idea to look at your fragment size before going into those downstream applications. And without process contaminants to the sample work purification, downstream modifications and assessments are more reliable. And it makes sense. Polymerases and restriction enzymes have preferred buffers in salts and do not like organic solvents. These factors can impact the thermodynamics of the reaction. Shown here is a GenElute E and a silica-based assay. And going back to the PCR itself, what you're looking for is earlier cycle rate, meaning that the reaction will happen faster or happen sooner. And that indicates that you have enough target sequence within your tube, enough to initiate the reaction. and that will help with the quantitation of it. So this could shift your quantitation of qPCR. And also, if you don't have enough sample in the tube going into this, due to the quantitation differences, you'll see a shift in the cycle count. Additionally, there can be inhibitors within the actual tube itself, your sample itself, coming off of each of the different sample preparation technologies that can inhibit those the reaction from happening itself. The E in GenElute E highlights several eco-friendly improvements. To start, there is no liquid waste associated with the GenElute E purification method. Typically, in a molecular workflow lab, you can essentially add a bleach to a biohazardous liquid to allow to decontaminate it. However, due to chaotropic salts being in solutions of silica-based, it can produce a toxic gas when combining chaotropic salts with a bleach solution. So you have to dispose of those liquid waste separately. The negative chromatography works completely different, where the sample that you load is the sample you get back. So this completely eliminates all of the liquid waste associated with the workflow itself. Additionally, we kept sustainable packaging in mind, where 70% the packaging itself is made of 70% recycled content. And this is the very first product that we have that is going to feature a starch-based compostable packaging. And this is what the columns are actually packaged in, as well as some of the other components for some of the higher throughput kits that are also available. And since there's fewer centrifugation steps, less energy is used, and this also, just thinking about sustainability from an efficiency aspect, it does increase sustainability by simplifying the workflow. And if you take the plastic waste itself, and this is the big one, if you take all the pipettes and tubes and the columns themselves, you weigh them separately between what's used for a silica-based workflow and compare it to the GenElute E Single Spin technology, you see a 55% reduction in the plastic weight associated with actually running the protocol, which is huge for the environment. Also, when considering your PCR workflow, please consider the GenElute E technology, where you can do your sample preparation using a GenElute E kit. Then just go right into PCR with whatever reagents that best fit your application. And then you can clean up your PCR with our DNA cleanup kits for further downstream assessments. The key applications that we are featuring this technology with to start of note are for whole blood, cell culture, tissue, and plant genomic DNA, and then cleanup based off of the impurity itself, such as those phenol components that can impact your sample, cleaning up primers and DNTPs post-PCR, and as well as dyes that may be added to the sample for other workflows as well. There are additionally some supporting products associated with this, which are the cap puncher, which allows for you to choose your own workflow, an FFPE deparafination solution, which is a xylene-free, so again, it's good for the environment, and a tissue stabilizer, and then we also have an RNA gel loading buffer. Thank you all for taking the time to listen to this webinar. I hope I helped you gain some insight into PCR and how sample preparation workflows play a role in their success.